A very big hello to all my wonderful PU2 and KCET champions. I hope every one of you is doing really good. Yes, so I welcome you all to our Masterstroke series, where today we are going to deal with the chapter Biotechnology and Its Applications. And for those of you who have not watched Biotechnology Principles and Processes, go watch it as well according to whatever time you have. And always understand that these videos are more than enough for your concept clarity and everything. And I'll be telling you what questions are going to come from these chapters as well. And let me tell you, if at all you have ever felt that this chapter is difficult, it's actually easy, and there are only few concepts, okay, but we misunderstand this chapter to be difficult. So I'll try my best to make sure that you also feel the same. Yes, and my dear children, do not forget to give your best every single time, and let's go ahead with this chapter right now. So the first thing that we have over here is tissue culture. What do you mean by that? So basically, suppose there is a very nice plant. This plant has all the good characters. It is pest resistant. It is this, it is that. Lots of good things are there. Now what we want to do is that we would want to make multiple copies of it. Okay, but how do we do that, ma'am? So there are actually certain cells which has an ability to divide, go on dividing and give rise to a whole new plant. You call these cells as meristematic cells, and mostly they are located at the apex. That means at the tip. Okay, so this part which you call this as apical meristem. Cool. Similarly, over here you might have seen there are some buds over here which can give rise to a branch or a new leaf and things like that. Basically, these buds that you see over here, you call them axillary meristem. So here also you have some cells which can go ahead and keep on dividing. These cells are known as meristematic cells, and depending upon their location you have different names. So basically I can take some cells from here or from here, wherever I want. Suppose I am taking some cells from here. Okay, now this part that I have taken from the plant is known as X-plant, because now it is not the part of the plant, it is an X basically. So this is what? An X-plant out there. Now what are you going to do with this X-plant, my dear children? You're going to put it in a medium that has a lot of nutrients. Why? Because you want to make sure that the cells over here divide and keeps on dividing. You want to make a new plant, right? Now when you get a bunch of cells like that, you will now not call it an X-plant, rather you give it a different name. You will now call this as a callus. Callus, yes, callus, okay. And then you put it in another medium which will be helping in the formation of root. So probably there might be some hormones that will be helping in the formation of roots. Then also it will form what? A shoot form of structure. First root, then shoot, everything is going to be formed. Why? Because you are putting in a medium that gives hormones needed for both what? Shooting as well as rooting. Got it? And now what do you get? You get small plants, my dear children. And these small plants, you can put it in actual soil and they will be growing up there. So basically what I am doing over here is that I am taking some tissue. Okay, a group of cells are there and I am making lots and lots of different plants, right? So this is known as tissue culture. Growing a whole plant from an X-plant, that is any plant in what? Sterile condition. You have to make sure there are no bacteria up there, that otherwise it could cause a problem. So over here there are sterile conditions being maintained. X-plants are placed in special nutrient medium for what? Growth. And you also need to understand one very important thing. Basically these cells have the ability to divide and give rise to a whole new plant, right? So this ability is known as totipotency. What do you call it? Totipotency. So ability of a single cell or an X-plant to regenerate into a whole plant is known as what? Totipotency. Not all cells has that ability. There are some specialized cells and they are known to be totipotent cell, and the ability is known as totipotency. Okay, now just think of this scenario where from just tiny X-plant I am making lots and lots of plants. So many of the plants, my dear children. Since I am able to make lots and lots of plants out there, this is known as micropropagation. What do I call it? Micropropagation. So basically with the help of the tissue culture method, what I am doing, I am propagating lots of organisms or lots of plants up there, and this is known as micropropagation, producing thousands of genetically identical plants rapidly. This production, when I'm doing it in a large scale, producing thousands of them, I'll call this as micropropagation. For this, I use a technique that is known as tissue culture. Now, you have to notice one very important thing. Basically, all these plants that are going to grow, my dear children, are from the same type of cells. That means genetically, if I say all of them are going to be same, isn't it? Yes, genetically, all these plants are going to be same, which is why I can call them clones. I can call them clones, but I have a specific name for them that is known as soma clones. That is known as soma clones. So there's a usual question that comes. What is the difference between somatic hybrids and soma clones? Or basically in MCQ, they might be asking you, what is the end product of micropropagation? Somatic hybrids or soma clones? Soma clones is the answer. We will soon see what you mean by somatic hybrids as well. Okay, so the clones that you get over here with the help of micropropagation where you use tissue culture method, this is known as the end product of this are known as what? Soma clones, because they are genetically very, very identical. Okay, got it. Yes. Now used for commercial mass production of crops like tomato, banana, apple, etc., etc. I hope this part of the story is clear. What is the end product of micropropagation? Soma clones. Okay. Now we'll go on to one very important question. Ma'am, if I want to produce virus-free plants, only virus-free plants, which part of the plant should I take? Always remember, you can have two options, either apical or axillary meristem. Ma'am, is this a question? Yes. This is a PU2 question. For production of virus-free plants, what is the part of the plant that you take? You take either apical meristem or you take axillary meristem. So virus-infected plants still have healthy meristems. Okay, so even if a whole plant is viral infected, their axillary and apical meristem can still be virus-free, which is why you only take those particular portions. Now, meristem culture allows growth of virus-free plants successfully used for crops like banana, sugarcane, as well as potato. That's a secondary thing, but at least remember this thing. Apical and axillary, do not forget it, okay? Yes, apical comes at the tip. It can be the shoot tip, it can be the root tip also, okay? And axillary is a place from which, you, as I said, you can get a branch, you can get a new leaf or things like that coming up there, okay? Yes, fine. Now, I told you, somatic hybrids and soma clones, both of them are different, right? Now we are going to see somatic hybridization. What is going to happen up over here and why do you call them somatic hybrids? So listen to me carefully. Suppose this is a plant cell. My dear children, plant cells has an extra wall layer. You call this as what? Cell wall. You will call this as cell wall, right? Now suppose I am removing the cell wall of a plant cell. Let's do that. Let's remove the cell wall of the plant cell. What is left behind? Just the cell membrane and all the cell content, right? I will call this as protoplast. I will call this as protoplast. So basically, when I remove the cell wall of a plant cell, now the remaining structure which is there, I'll call this as protoplast. Similarly, suppose I take another plant cell in which also I have removed the cell wall and then I make both of them to fuse. Basically, I make the protoplast of both of them to fuse. This is known as protoplasmic fusion. What do I call it? Protoplasmic fusion, or you call this as somatic hybridization. You're getting a new hybrid out there, new thing out there. That's why you call it hybrid. It was not generally found. Okay, so first of all, cell walls of the plant cells are digested to obtain naked protoplast. Cell wall is being digested with the help of cellulase and things like that. And lastly, you get protoplast, the naked protoplast. Fused on the protoplast. Protoplast from different plant varieties with desirable traits are fused. Suppose this particular tomato plant has a very good pest resistance. This particular potato plant, this is tomato, this is potato, okay? It has very good drought resistance. So basically, I have their desirable features and making them fuse together. So what do I get at the end? I get hybrid. So fused protoplast develop into a new hybrid plant. These are known as somatic hybrids. The resulting plants are known as somatic hybrids. Example, pomato. Pomato is what? Potato plus tomato is what you get as pomato. So over here, you get the end result as somatic hybrids. Remember, they are different. It was not the same as in the parent plant. Parent plant were different. The progeny that you get at the end is also going to be different. There, soma clones in micropropagation, you are basically producing same type of plants. Here, different type of plants. So please remember, protoplasmic fusion ends up in somatic hybrid formation. Micropropagation ends up in soma clone formation. There's no need to mug up, just use your little brain out there. Yes, that's all. Yes. So pomato, please remember the example of that, and that's all. That is somatic hybridization. Okay, Okay. So now you're seeing that a lot of things are happening up there. You are bringing in a lot of science into agriculture, right? Similarly, one very important thing happened, and that is genetic modification of organisms. Basically, you went straight inside the genes, and suppose you have a good gene for this particular character, you took that gene and placed in some other organism and saw it worked as well. That is known as formation of genetically modified crops. So we studied in biotechnology principle and processes how to make a recombinant DNA and things like that. The same thing is over here. We are basically producing recombinant plants out there in this particular chapter. Not just plants, we will see a lot of things also. So anyway, first we'll talk about plants. Okay, so whenever you have a new DNA being inserted, that means it is modified. It is genetically modified, which is why we call these organisms as what? Genetically modified organisms, or GMO. And in the case of plants, they have so many benefits, and this is a usual PU2 question as well. Okay, what are the benefits of genetically modified plants? So let's see one by one. The first benefit that you have over here is abiotic stress tolerance. That means if in a plant I'm putting some gene which will provide it tolerance to what? Tolerance to cold, tolerance to drought condition. Without water, without much water also, the plant could be able to survive if at all I put a gene into that which will provide it stress tolerance. Okay, so similarly it can resist to high amount of salt, high amount of heat and things like that. So the first good thing about GM crops or genetically modified crops is what? It basically provides abiotic stress tolerance. The second good thing, pest resistance. So suppose I have a gene which provides resistance to some pest. I put in that particular gene into the plant, and now the plant is safe from all the different type of pests, right? So pest resistance is another thing. And under pest resistance, my dear children, you see two examples. One is Bt cotton, about which we will be studying first, and then we will talk about a method that is known as RNA interference. Do not be worried, it's simple. Okay, both of them provides pest resistance. So Bt cotton and RNA interference, you also call this as RNAi. RNA interference is also known as RNAi for interference. Got it? Yes. So anyway, the end results, what do you get? You get basically the pest resistant plant. So that is the next feature that is offered by genetically modified plants. Next thing, reduced post harvest loss. That means most of the time it happens that we harvest the plants, the farmers, they harvest the plants, but later on there are so many losses when you keep it under go down, my dear children, that is fungi, this and that being formed, and all of it leads to loss. So here in genetically modified plants, you will also be able to see that there is reduced post harvest loss. It improves the storage as well as the shelf life, as in you can keep it safe for a very longer time. That is what you mean by shelf life. Okay, yes. Now efficient mineral usage, that is the next thing. That means a plant which is genetically modified. Okay, suppose it was staying in a very nice environment where you had so much amount of nutrients and things like that, it could sustain. But generally the plants, if at all you're putting it in a place where there are less nutrients, it usually dies up, right? But in the case of genetically modified plants, suppose it is a genetically modified plant, this one, what is going to happen? Even with little amount of nutrients, okay, even with little amount of nutrients, it can survive. So efficient mineral usage means prevents rapid soil fertility loss. So here, my dear children, what do? Even if they have high amount of nutrients up there, they will be taking up only a few. They will be taking up only a few within, because within this few, they can easily survive. That means basically, they are making sure that the soil also has not lost the fertility. The soil also have enough nutrients, and that is a good thing, right? So efficient mineral usage is another point. First thing, abiotic stress tolerance. Okay, pest resistance is the second thing. Third one, reduced post-harvest losses. And fourth thing that you have is what? Efficient mineral usage. And last but not the least, enhanced nutrition. So you know what? In places like Africa and all, people were dying of malnutrition. Okay, people were dying of malnutrition, and the staple food that they had was rice. They used to have rice. So then, scientists thought, why not make this rice have some enough amount of nutrients such that people who have it at least get those nutrients as well. And that's how now exactly not that's how, but still you can remember it like that. That's how golden rice was being formed. So what do you do in golden rice? What is so special? Why do you call it golden rice? So basically, the gene which is there for production of vitamin A, and that is being inserted into the plant, which is why the rice which is going to be produced also will have vitamin A, and that was used for what do you call taking down the problems that were caused due to what? Vitamin A deficiency. So vitamin A deficiency was tackled by the production of what? Golden rice. So what are you doing over here? You are enhancing the nutrition quality of what? Of the plant by putting in some genes. And the example for this is golden rice. Always remember, it is vitamin A enriched, which is why it is giving a goldenish, yellowish golden color out there. Got it? Yes. So people who are having rice as staple food, they could also get this vitamin A, which is going to help them in improving their eyesight and things like that. Got it? So these are the few things that you need to remember. This is a usual question that comes in from this particular chapter. Have this in your head. Okay, so is there any other mechanism for production of better crops? Yes, genetically modified crops as the best way out there, that is you use recombinant DNA technology. Now we are going to see examples. How are you going to make a plant pest resistant? That is the first thing that we are going to deal with. And for that, the first thing that we have is production of Bt crops. What do you mean by Bt? Bt stands for Bacillus thuringiensis. It is actually a bacteria. Okay, what is spelling of it? Bacillus thuringiensis is going to spell like this, my dear children. And you also call this as biopesticide. So basically, remember, you use chemicals on the plant such that no pest will be eating it up. Similarly, here an organism itself is going to kill the pest, which is why you call this as biopesticide. You're not using chemicals, you're using an organism that could kill the pest. You might wonder how it is making the plant pest resistant. So let me talk about that right now. So here, my dear children, I have my chutku Bacillus thuringiensis. In Bacillus thuringiensis genetic material, there is a gene, my dear children, which you call as cry gene. And you know, genes are responsible for production of what? Proteins. So here also a protein is being produced. But this protein which is being produced over here is inactive in nature. It is not active, it is inactive in nature. Always remember that. This protein which is there, it is actually a toxic substance out there. But this, since it is inactive, which is why you will now call this as a protoxin. That means it is not the active version of toxin, it is just a protoxin. Now, to make this particular toxin active, what do you need? You need some proper environment. And where do you get that? I'm going to talk about that over here. So suppose a caterpillar, okay, a caterpillar is there, and this particular caterpillar eats up this bacteria somehow. Probably this bacteria might be there on the leaves and things like that. Suppose it's there. Okay, this bacteria is very small. I have made a bigger picture, but it is actually very small. The bacteria reaches up over here. The bacteria, suppose it reaches up over here in the gut of the caterpillar. Suppose the mid-gut region of the caterpillar. What are the changes that you observe in the mid-gut region? The pH over here, the pH over here is, my dear children, highly alkaline in nature. That means it is basic in nature. Now what happens? In this alkaline pH, the bacteria's protoxin which was there, it actually converts into the active form of toxin. So it was protoxin until now, which was inactive, and now it is going to convert into what? The active toxin. And what is this active toxin going to do? This active toxin is going to mess up with the epithelial cells, that is the lining, okay, epithelial cells of the mid-gut region, and later it can lead to perforation as well. That means formation of pores as well, and finally it could lead to the death of this particular organism out there. Got it? So mid-gut, in the mid-gut is lined by the epithelial cells. These epithelial cells are being targeted by this active toxin, leading to what, my dear children? Leading to even perforation, that means formation of pores as well, and finally leading to death of this particular organism. This organism, wasn't it eating the plant? Yes. So wasn't it a pest? Yes. Aren't you making the plant pest resistant if at all you're transferring this gene inside. Didn't get it? Listen to me. Until now, it was the bacteria who was doing it. Suppose we take the gene from this particular bacteria, this cry gene from this particular bacteria, and put it inside the plant. Put it inside the plant. The plant will now produce the protoxin. So every time a caterpillar comes and eat the plant, what is going to happen? The caterpillar is going to die. That is why we say that it is providing pest resistance to, this particular gene is providing pest resistance to the plant. And here, since you are taking the genes from Bt, okay, that is a Bacillus thuringiensis, suppose you are inserting in a cotton plant, you will now call this as Bt cotton. Why? Because you have taken the gene from the Bt and you're putting inside a cotton plant. That is why you call this as Bt cotton. Got it? Okay, and always remember, we have studied in the previous chapter also, whenever we want to transfer something into the plant cell, we need a vector. We need a vector, and usually that vector is Agrobacterium tumefaciens. This is another bacteria. Agrobacterium tumefaciens. This is another bacteria which has, my dear children, a plasmid, an extra chromosomal thing out there that is known as plasmid. Here in this particular plasmid, you have a region that is known as transfer DNA, which is actually going to be transferred into the plant and causes tumor in the plant. But now, since we do not want to transfer that particular portion, we remove that and we put our gene of interest. What is our gene of interest over here now? Cry gene. So we put the cry gene. This is the cry gene, and then this particular Agrobacterium tumefaciens goes and infects the plant, that is suppose a cotton plant. Now what will it transfer? It will transfer the cry gene out there, and tada, we have got the cry gene in the plant. So you need a vector, please remember that. In the case of plants, it is usually Agrobacterium tumefaciens which is going to act like vector. What is Bt? It is Bacillus thuringiensis, a bacteria that has cry gene. Remember, the cry gene is responsible for production of what? A protein. The inactive form of protein is known as protoxin. Okay, now later when the bacteria has been taken up by a caterpillar or any other insect, my dear children, what is going to happen? It reaches the midgut region. There the pH is alkaline. In this alkaline pH, the protoxin, the inactive version of toxin, converts into active toxin and leads to what you call damage of the epithelial cells, and later leads to perforation and finally the death of the organism. Okay, now we suppose we are using the same mechanism. In the case of genetic modification, what do? We take the gene from here, we put it into the Agrobacterium tumefaciens, and the Agrobacterium tumefaciens is going to infect the plant, and basically while it infects, what does it pass? It passes the cry gene, and tada, the plant is now pest resistant. It can produce its own toxins. Protoxins are there. Works? Works. Now listen to me very, very carefully. We will just read out whatever we have studied until now, and then we will go ahead with the new things, okay? So, produced by Bacillus thuringiensis and used as biopesticides, genetically modified to resist insects, example Bt cotton, Bt corn, Bt rice, Bt tomato, Bt potato, Bt soybean. So basically, you're just putting the gene into these plants: corn, cotton, rice, tomato, potato and soybean, etc. Okay, now Bt produces insecticidal protein. What do you mean by insecticidal? As in insect killing protein out there. Target specific insect pest. Toxin exists as inactive protoxin. I told you, right? Does not harm the bacteria. So why it does not harm the bacteria? Question. Because it is in the inactive form. In the inactive form, you call this as protoxin form. Now, insects ingest the protoxin, as in basically insects which are there whenever they eat up the bacteria unknowingly, my dear children. What happens? The protoxin gets activated in the alkaline gut pH, and the activated toxin binds to the midgut epithelial cells, creates pores out there and leading to cell lysis and insect death. The cell is going to be lysed, that means it is going to be cleaved open and leads to the death of the insect as well. Now, the cry genes which are there, they are very much specific to certain insects. That means this gene codes for this protoxin which is only going to kill this insect. That is what you mean by what do you call being very, very specific. Okay, so there are different forms of cry gene. 3 of them are there and 3 of them you need to remember. And there is an easy way to remember this. Okay, let's try it out. Listen to me carefully. This is also a question. In KCET, this is a question. In PU2 also, this is a question. Okay, so we put cry, we put cry and we put cry. This is going to be, okay, listen. So cry 1AC, okay, cry 1 AC. This is one type of gene that you have. After one you have two, right? So, and after this you have AB. You have what? AB. So AC comes first, AB comes second. Fine. Listen to me. The first letter of the organism starts with C. First letter of the organism starts with C. So can I write cotton? Yes, I'll write cotton. So first letter of this organism starts with C. Second letter of this organism starts with B. Second letter is what? Or the second word of this particular organism starts from B. So over here you have B. Why I am telling you this is for a reason. Now over here you have one more gene. So Cry1AC is done, Cry2AB is done. Where is Cry1AB? So let's write Cry1AB over here. Here there is only one. The name of this organism is corn borer. Over here, first letter with C, second letter with B, corn borers, and there is only one gene which is responsible for producing toxin that can kill the corn borers. So two genes for cotton bollworms, one gene for corn borers, and over here both of them starts with C and B only. But remember, cotton bollworm is a bigger name, which is why you need two genes, and corn borer is a small name, which is why you just need one gene. Okay, so why I have told you the first letter starts with, remember like this, because mostly what happens is that we write 2 AC there and 1 AB over here. Don't do that mistake. Okay, so first letter starts with C, cotton. First word starts with C and the second word starts with B. Second word starts with B. This is cotton bollworm when you have two things. Okay, now over here Cry1AB, only one is there because this name is also small. First letter starts with C and last letter ends with B. Here also I do not want you to get confused. Don't write 2 AC and things like that. There is no 2 AC. There is only one AC, and AC comes up there for cotton bollworms only. Okay, so these genes are going to target cotton bollworms. This gene is going to target corn borer. That's all. Okay, I know it's a little bit confusing, but somehow you will be coming into a loop for that. Yeah? Yes. Now try it out by yourself. Otherwise, you might forget it. Okay. Now, different cry genes target specific insect groups. So you should know the insect groups as well. Okay. So first one is Lepidopterans. In Lepidopterans comes tobacco budworm as well as armyworm. Then you have Coleopterans. In Coleopterans comes beetles. Dipterans, in that you have flies and the mosquito. So flies and mosquito in Dipterans. Coleopterans have beetles. Lepidopterans are basically tobacco budworm as well as armyworm. Just remember 3 groups of organisms: Lepidopterans, Coleopterans, Dipterans. Okay. And the thing that I have told, this is all about the BT cotton, or how you use biotechnology in the case of agriculture, how you produce a pest resistant plant, okay, using Bacillus thuringiensis. Now you have one more thing. I already told you, you have one more very, very important thing, that is RNA interference method. RNA interference method, this is also easy. Okay, listen to me, what usually happens. So generally, my dear children, you have the DNA. DNA is double-stranded. From the DNA undergoes transcription, right? Transcription, what do you get? You get RNA, that is mRNA. And that is single-stranded structure. It is single-stranded, please remember. It is single-stranded. Now this single-stranded structure which is there, it is going to undergo another process that is known as translation. And at the end of translation, what do you get? You get protein. So basically, for production of protein, you will need a single-stranded mRNA. Okay. Now, suppose an organism is there. The name of the organism is Meloidigoin incognita. It is basically a Aschelminthus, a type of worm that you have, but they are coming under nematodes, Aschelminths. Okay. Now, this Meloidigoin incognita, I call it as MI. It goes and infects the tobacco plant. It gets into the tobacco plant, it produces its own mRNA such that its own proteins are being made, and these proteins are very, very bad for the tobacco plant. Now we do not want the organism to affect the plant. What can be done? We can stop the production of this particular protein, but how can we do that? Basically, there is only one method. Let's make this single guy mingle. So that means if at all there is one more strand coming up right over here which is complementary to this mRNA. What do you think? Will it undergo translation? Because now it is not single stranded anymore, it is double stranded. Do you think translation process can happen? No, it is not going to happen. So aren't you interfering in the RNA translation process? Yes, which is why you call this as RNA interference. This is the logic. You are going to interfere in the translation process, my dear children, and for that you need one more RNA strand. But where do you get that from? Listen to me carefully. So what exactly happens is that we get to understand the genes which are there in the nematode. Okay, in this particular nematode, whatever gene is there responsible for formation of this particular mRNA, we get to know that. Okay. Now this gene, which is very, very important for us, is being put into the tobacco plant. Into the tobacco plant. But can it be put directly? No. Again, you need a vector. What is a vector over here? I told you it is going to be Agrobacterium tumefaciens. Here I have the plasmid. In the plasmid, I am going to put the gene right over here which I have taken from this organism. This gene is not going to form the protein. Do not be worried. It is not going to form the protein. It is going to form something else. I am telling you. So I have taken the gene that is going to produce double-stranded RNA. I have taken the gene that is going to produce a double-stranded RNA, which you also call as dsRNA. Okay, so this gene is going to code for double-stranded RNA inside the plant. Okay, it is not going to code for the protein. Do not be thinking that why I am putting the protein that is toxin for the plant here itself. No. So basically, I took this gene, I took the Agrobacterium tumefaciens, it went and infected the plant, and now the plant has the gene that coded for the double-stranded RNA. This double-stranded RNA which is going to be formed, my dear children, is very, very important for us. Why? Because the nematodes, suppose the nematode tried to infect it, and the nematode's mRNA is right up over here. The single-stranded mRNA is right up over here, which is going to undergo the process of translation to produce the protein. I am going to hinder in its activity. I am going to stop that activity that is going to happen. What do? This particular double-stranded RNA comes over here. Okay, it reaches up over here. First thing that happens is it cleaves only one strand. Okay, out of this double-stranded, only one strand comes over here and attaches. What happens to the other strand? The other strand is not coming. Just remember, it's just right there. It's just right there. You just need one strand from the double-stranded RNA. You got one strand that attached over here. Do you think translation will happen? No. Do you think protein are going to be made? No. Aren't you interfering? Yes. How? With the help of the gene that you took from the nematode itself. This gene produced what? Double-stranded RNA. So you're interfering with the help of the double-stranded RNA. Do not go and write single-stranded RNA. For this particular thing, you require a double-stranded RNA itself. From this double-stranded RNA, only one strand goes up there and binds. But you require double-stranded RNA. If at all you write single-stranded RNA, your marks is going to be cut. Got it? Got it? So what did you do, my dear children? You took a gene from the nematode with the help of Agrobacterium tumefaciens. You put it in the plant. In the plant, that gene coded for double-stranded RNA. This double-stranded RNA was taken to the place where mRNA was being made already. This double-stranded RNA cleaved. Only one strand went and bound with this. Now it is not single-stranded anymore. It will be double-stranded. The mRNA is going to be double-stranded. Later, it is not going to undergo process of translation. It cannot produce the protein which is required for the plants to die. So which is why the plant is absolutely safe. This is known as RNA interference, and as I said, you also call this as RNAi method. Fine, simple. Now over here, let's read it out and then all things will be even more clear. So Nematode problem. Meloidigoin incognita infects the tobacco roots, reducing its yield. So where does it affect? The roots of the tobacco plant is the affected place. Now RNAi strategy. Silences specific mRNA to prevent nematode infestation. What does it do? It silences. How does it silence? By going and binding up there. What does it silence? The mRNA out there, because you do not want it to produce the protein. Okay. Now RNAi takes place in all eukaryotic organisms as a method of cellular defense. So this is very, very important. RNA interference actually happens in eukaryotes. Eukaryotes are basically what? The ones which are more advanced. Our cell, we are eukaryotes, my dear children. Our cell will be having all the proper things, that is our nucleus is there with a nuclear bound membrane, there are membrane bound organelles, lot of things. There are prokaryotes, for example bacteria, they do not have any of the nucleus and things like that. They're very primitive in nature. So in the case of eukaryotes, only in the case of eukaryotes, this is a question that comes. They write that RNA interference takes place in prokaryotes and eukaryotes as a method of cellular defense. Wrong. Only in eukaryotes it acts as a cellular defense. Always remember that. Now mechanism for RNA interference. Double-stranded RNA formation. Complementary RNA binds to target mRNA, blocking the translation. What do you need for that? You need a double-stranded RNA. Now agrobacterium vector is used to introduce the nematode specific genes. So I use an agrobacterial vector for putting the genes inside, or you call the plant. Now host plant produces sense and antisense RNA, forming double-stranded RNA. As I told you, one RNA, another strand. So sense and antisense is what you call it. Fine. Now double-stranded RNA triggers RNA interference, silencing the nematode mRNA. It does not allow the mRNA to undergo translation. Result, nematode cannot survive, making the transgenic plant what? Resistant. Now here I use the term transgenic. Transgenic means what? Modified plants. Genetically modified plants are known as transgenic plants. Genetically modified organisms, animals, are known as transgenic animals. So over here at the end you get genetically modified organism, or you also call this as transgenic organism out there, or a plant out there. That's all. This is RNA interference. Always remember, you need double-stranded RNA. It works in the case of eukaryotic cell. Okay, it's a method of cellular defense in the case of eukaryotes, not in the case of prokaryotes. And here, this meloidigoin incognita, where is it affecting? It is affecting the tobacco roots. Cool. And this is the picture that is given up over here in your NCERT. That's all. That's all about biotechnology in the field of agriculture. Now we are going to go ahead, we will see what is the use of biotechnology in the field of medicine as well. There are two examples for this as well. One is insulin production and the second one is gene therapy. Okay, we'll go one by one, we'll study that. Until now, if at all you think it's clear, first of all, pause the video. You do not need to listen to me again because I know it's boring. You just need to read through the paragraph. You can stop the video and use your NCERT to read through the paragraph from your NCERT. Each and every line is important. Come back, start off this video again, and let's go ahead with insulin production. Okay? And if you feel that whatever I am teaching you're able to understand it, do let me know in the comment section, and do not forget to hit the subscribe button, my dear children, as well. Okay? Okay, fine. Now we talk about insulin production. So what happens is that generally, okay, before that, just a second. We have our pancreas. I hope you guys know that we have our pancreas, right? What does the pancreas produce, my dear children? Pancreas are there for the production of insulin. Okay, what does the insulin do, my dear children? In our blood, suppose there is too much amount of glucose molecules. It's a problem. Because the blood sugar level, this glucose is known as what? Sugar. The blood sugar level is high. What do you need to do? You need to trap this glucose and convert it into something else which you call as glycogen. You need to convert the glucose into glycogen. So many glucose come together to form a glycogen, and this glycogen can be stored in the liver, and tada! Who does that? It is insulin who does that. Now suppose in some people, the pancreas are not working really well, which is why there is no production of insulin also that is happening. Insulin production is also very less. So what can be done to maintain this thing to happen? To make sure that this thing is happening, conversion of blood sugar level to glucose into glycogen is happening. You can only do one thing, and that is to give insulin from outside source. So initially, the different cattle such as cows, or for example pigs, insulin was being used, okay, for treating the condition that is known as diabetes mellitus. But my dear children, later on people started having allergies, and it was understood that okay, this is not the correct way to do it. Probably we have to do something else. Okay. Then came a company whose name was Eli Lilly. This is a very famous company, it's still there. Okay, it is known as Eli Lilly. What did this company do? So, before I tell about that, you should know the basic structure of how insulin looks. So, you have multiple chains. This is a protein thing that you have. It's a hormone, but it's a proteinaceous hormone. It is made up of protein molecules. So, there is an A polypeptide, there is B polypeptide, and between that you have a chain that is known as C polypeptide. Okay? Listen to me very carefully. This is pro-hormone. What do you mean by pro? You remember protoxin? That means it was inactive. Similarly, when I say that a hormone is pro-hormone, I mean that it is not mature form of hormone. This is not the working form of hormone that we have. To make this hormone work, in the case of us, that is the eukaryotes, my dear children, you have one very important thing which is going to cut off this C peptide. The C peptide, whenever it is going to be removed, now you get A and B, and both of them are linked with the help of some disulfide bonds. Now you will be calling this as what? The mature form of hormone. So this is the mature hormone that you have. So there is pro-hormone and there is mature form of hormone. In the pro-hormone, you have the C peptide. In the mature form, you do not have the C peptide. Does it happen naturally in our body? Yes. In the case of eukaryotes, there are enzymes that is going to cut off this off, and it is all going to occur very normally in our body. Okay. So, Eli Lilly had a plan of production of insulin in E. coli. E. coli is a bacteria. E. coli is a bacteria. Now suppose I take the gene for insulin and I put it inside the E. coli. What is going to happen? In the E. coli also A, C and B chain are going to be formed. That means only we will get the pro-hormone form. We will not get the mature hormone form. Because E. coli is a prokaryote, it does not have the gene or the enzyme for cutting the C peptide, right? So you will get only the immature or the pro-hormone form, and that is not something that we need. That was a very big problem. Okay, that was a very big problem. So to tackle that, Eli Lilly came up with an idea. Why not produce only A chain over here and B chain over here? But how can I do that? So the gene which is coding for insulin, why not take the gene that is coding for A and that is coding for B and put it in two organisms out there? So here A chain is going to be produced, here B chain is going to be produced. Then when it is being formed, you can take the A and B chain, and in the laboratory condition, why not make the sulfide bonds? That is pretty much easy, and that's how you get the insulin, right? This could be done, and that was exactly how humulin was being formed. Human insulin in an organism was being formed. This is known as humulin. This is known as humulin. So the genes were taken from human beings itself. Okay, our own A and B peptide are being produced separately, my dear children, and they're taken up in a laboratory condition. They are joined up together for the production of humulin. This was the whole idea. If at all you want, please take a screenshot of this. This is very much important. I'll just repeat. So whenever our pancreas are not producing enough amount of insulin, what is going to happen? The blood sugar level is going to increase, and consistently if this is happening, you say that you have diabetes, right? Now to tackle that, initially we used the insulin from some cattle or pigs, but later it was causing some allergies, which is exactly why Eli Lilly came into the picture. They said, why not produce the A and B chains of insulin separately in the E. coli and then later join them with its sulfide bonds. That's how you will get the mature form of insulin as well. Before that, just have an idea that in our case, A, C and B chains are actually being produced. The C chain is being removed, and that's how you just get the A and B. When you have the C chain, you call this as pro-hormone. This is not mature form of hormone, and when you remove the C chain, you get the mature hormone out there. Got it? That's all. Okay, this is the whole funda that we have. Very, very important. Okay, Now we'll read it out. So, earlier source insulin was extracted from cattle and pig pancreas. The problem was animal insulin caused allergies and also immune reactions. Cool. Human insulin structure consists of two polypeptide chains, A and B, linked by what? Disulfide bonds. There are disulfide bonds out there. It looks like this. So, this is the A chain. This is the B chain, and here you have the disulfide bonds out there. This is what the mature form. Okay, immature form, as I said, the pro-insulin or the pro-hormone form is where you have A and B chain, but you also have a C chain in middle. You also have a C chain. So A and B chain are having disulfide bonds here also, but there's a C chain. You have to remove the C chain out there. Okay, you have to remove the C chain out there. So human insulin structure consists of two polypeptide chains. I told you, pro-hormone initially produced with an extra C peptide, which is removed during maturation in our body, in our normal cells itself. Challenge in our DNA technology, that is recombinant DNA technology, assembling mature insulin from separate chains. That was very, very difficult. So that is how solution came into the picture. Eli Lilly created two DNA sequences for A and B chains of the human insulin. A and B chains separately, we got two DNA sequences, introduced them into E. coli plasmids for separate production, extracted them and combined the A and B chains using what? Disulfide bond formation, and that is how large-scale production of human insulin using bacteria was done, avoiding any sort of immune infection. Why we are digital? Because it's our own type of human insulin that is being produced, right? And which is what you call as humulin. Oh, sorry, I gave the spelling as Humilin. It is humulin. Okay, humulin. It's not humulin. It's humulin. Just make that correction. Cool. So always remember that. So here you have A. I'll make the diagram and I'll show. Make B also. In between both of them, just join something like this. So A and B chain are joined with the help of a C peptide. Okay. Here you already had some bonds. This is the pro form of insulin. You call this as pro-insulin. Okay? Yes. That's all. Remember, it's a hormone. You mention this as a hormone. Do not mention this as a pro-protein. No, it is not a pro-protein. Mention this as a hormone, even though it's made of protein only. But since it is a hormone, you call this as pro-hormone only. Okay? Yes. Now, everyone who is preparing for KCET examination, we have officially launched the KCET batch, where you have it is a comprehensive 30-day online crash course. Do not worry, it's not starting now. It is going to start after your boards. So it is going to start by 21st of March 2026. There's complete syllabus coverage, both your PU1 and PU2, and there will be KCET previous year questions along with their solutions. And if at all you want to give it in a test format, that is also going to be provided. Okay, there will be chapter-wise KCET in the test format with the proper timings and everything, and along with that you have 10 full-length KCET mock test as well. The pricing for this is rupees 2499. Remember, it's offer price. It is going to end by 25th of February. So if at all you're planning, you can take it up. And also, if you want a free mock test out there just for trying out, not mock test, mock test basically, PDF of a mock test, then basically you can go ahead in the link, the discussion session, my dear children, you will be finding a link. Fill in your mobile number and that's how you get the PDF. So yes, that all depends upon you. If at all you are very thorough with PU2, probably you can go ahead and do that particular thing. If not, you can take some time off and then later on you can try it out. Yeah, but KC30, please register for it. As I said, the timings for it, the offer price is going to end by 25th of February. Yes. Now, biotechnology in medicine, one more example was there, I told you, and that is gene therapy. What do you mean by gene therapy? So, my dear children, a child was there, and this particular child was a 4-year-old child, and she, in her cells, one particular gene which was there, that was not there. Gene which was there is not there. Oh my God, sorry, listen to me carefully. The gene which was supposed to be there, it is not there. Why? Because this got deleted. So I say, because of deletion of one particular gene, you had a deficiency of something. This gene was actually responsible for formation of adenosine deaminase. And whenever the term ACE comes in, you need to understand it's an enzyme. It's what, my dear children? An enzyme. So basically, the gene that was coding for adenosine deaminase got deleted, which means that there is no enzyme being produced. There is no enzyme being produced. Ma'am, is that a problem? Yes. Why? Because adenosine deaminase is responsible for detoxification of some substance inside the cell. So usually it is found in all the cells, but majorly it is super important in one of our immune cells, which you call as lymphocytes. So in all the cells they are present, but in lymphocytes they are very, very important. Lymphocytes cannot afford building up of any toxic substance. And who is responsible for making sure it's very clear? Adenosine deaminase. But suppose this gene is not there, do you think adenosine deaminase is going to be produced? No. Which is why there is a buildup of toxic substance. And lymphocyte which is there, it is what? An immune cell. And if at all an immune cell is affected, our immune system is going to be affected. If our immune system is affected, do you think we can live? No. It will later lead to death. So that is a very big problem. So what can be done? That is the next question. Okay, one thing that can be done is bone marrow transplantation. Why bone marrow transplantation? My dear children, in bone marrow, we have red marrow and we have yellow marrow. The red marrow have some cells which you call as hematopoietic stem cells, as in some stem cells which can give rise to blood cells, and under blood cells comes your lymphocytes and things like that. So from someone else we can probably take up a bone marrow, we can, with the help of someone else, we can do the bone marrow transplantation. The thing is that not always it would be working up, and sometimes, you know, the pre-existing cells are still there. The pre-existing normal lymphocytes which were there, we didn't have the genes, they are also still there. So which is why there is no coordination that is happening. There are a lot of troubles that is taking place. That is highly a reason why it is not always curative. Even though people have bone marrow transplantation, sometimes it can get rejected, and only the blood cells are being formed. What about the normal cells? They also need adenosine deaminase, my dear children. So in that case, that is not completely curative is what I would say. Okay, the second thing that you can do is what? Enzyme replacement therapy. You can take repeated transfusion of this enzyme, that is adenosine deaminase. You can insert it in an organism. But the problem with that is what, my dear children? Again, you need repeated transfusion. So that is also not, what do you call, completely curative. Okay, then what could be done? Then there is one more very important thing that is known as gene therapy. Okay, that is known as gene therapy. Now, if at all you do in a stage when it is embryonic, okay, when the baby is not just being formed, it's an embryo stage. At that particular point itself, you get to understand that this particular child does not have the gene, and at that same time itself, if at all you perform the gene therapy, it is all the more good. Why? Because it is going to be permanently curative, because you are putting in the embryo itself. So basically in an embryo, if at all you have seen, one cell divides multiple times, making clones basically, right? So if at all it has one cell has the gene, it will pass on the gene to all the cell. It will replicate and pass on the gene to all the cells. This is in the case of embryo. So if at all you are doing the gene therapy in the embryonical stage itself, it's all good. It is going to be a permanent cure. But if at all you are doing in the adult stage, again you will need repeated transfusion. But anyway, you should know how does gene therapy work. So I'm going to explain about that. Listen to me very, very carefully. So the first thing that you need to do is you will need to take the white blood cells, that is the lymphocytes, from the patient. You are going to take some lymphocytes from the patient. So we'll go one by one. Listen to me carefully. The steps of gene therapy is being asked as a KCET PYQ. Okay, and the steps of gene therapy is being asked in your PU2 question also. So over here, first thing that you do is you collect the lymphocytes. So collection of lymphocytes. Done. From what? From the patient. Now basically you culture it. Culture the lymphocytes. Okay, you make sure that there is an increase in number of the lymphocytes. Later on, what you're going to do, with the help of recombinant DNA technology, within the lymphocytes, my dear children, you are going to transfer the gene. The gene for what? Adenosine deaminase. Now, how do you transfer the gene in the lymphocytes? In the case of plants, we study there is agrobacterial tuberficins. In the case of animals, what do? You need a vector over here, and this particular vector, my dear children, is going to be known as retrovirus. In the case of animals, always remember the vector that you take is retroviruses, such as in HIV is also a retrovirus, but you do not take HIV virus. Remember, you're going to take something, okay, and whenever you take a virus, you will make sure that we are removing the portion that is going to cause us harm. So basically we say it is a disarmed retrovirus. Disarmed retrovirus. It does not have anything sword or anything like that. This is disarmed, completely disarmed retrovirus. It does just have the gene which has to be inserted. The gene is inserted. Okay. Now what do you do basically? You take this lymphocytes and put it back into the patient. So first you took them, you cultured them, you increase the number, then you put the gene into the lymphocytes using a vector that is known as retrovirus, and later you put it back into the organism. The thing is that after some point of time, obviously these lymphocytes are not going to live forever, they will be dying, which is why you will need repeated transfusion. So this is the case if at all you're doing this in adult stage. But as I said, gene therapy can be performed in embryological stage. If at all you perform in that particular stage, the organism will live. Okay, that is a permanent cure. This is not a permanent cure, you need repeated transfusion to happen. Got it? Yes. And always remember, what is the segment that you are putting in? You actually call this as cDNA. So as I told you, I need you to understand one very important thing. You have DNA. In the DNA, you have regions that codes for things, you call them as exons. They code for proteins, okay? And then you have regions that do not code for anything. You call this as introns. So when the process of transcription happens and a new RNA is being formed, the exons and the introns, everything are transcribed as such. You call this as hnRNA. This is the first product of transcription. Later, what do? You are going to remove the introns. The non-coding regions are going to be removed, and now you will just have the exons. You will just have the exons, and this is what you call as mRNA. A strand that just has the exon that code for proteins is known as an mRNA. Right. So if I put this particular DNA, what is going to happen? There will be coding, there will be non-coding region, lots of things. So why do you need to put that particular DNA? There is one thing that you can do, that is, you had the mRNA. Okay, mRNA has a sequence of DNA itself. Basically, why not do the reverse of transcription? So there was transcription where you made DNA to mRNA, that is known as transcription. If at all you reverse this reaction, RNA to DNA is known as reverse transcription, and over here you will be only having the DNA that has what? The coding sequences. You call this as cDNA, the complementary DNA. Complementary to what? The mRNA. You do reverse transcription, my dear children. You make the DNA. This DNA is known as cDNA, that is complementary DNA. That's all. And this complementary DNA is what you're going to insert over here into each cells. So please remember the flow. Usually we get confused. We think that culture comes afterwards. No, culture comes first according to your NCERT. So you have to remember it like that only. Okay, done. So this is how it goes. Now, I'll come back over here. Yes. Lymphocytes, white blood cells, are taken from the patient. Functional ADA-cDNA, ADA means adenosine deaminase, complementary DNA is introduced using a retroviral vector. Modified cells return to the patients. Modified cells are returned to the patients. And then remember, temporary, as lymphocytes are not immortal. They are going to die, which is why you need repeated transfusion. Before that, as I told you, over here you need to add one point, and that is culture the lymphocytes. Culture the lymphocytes. After extracting, culture them. Now, permanent cure possibility, introducing ADA gene into the embryonic cell for lifelong correction. That is the only way. So, bone marrow transplantation is not always curative. Similarly, enzyme replacement, again you need repeated transfusion. Gene therapy in adult stage, again you might need repeated transfusion, but if at all you're doing in embryonical stage itself, permanent cure. Got it? So there is only one permanent cure, and that is this. Fine. So it was first clinical use was in 1990, and it was done to a 4-year-old girl, and she's still alive, she's still there, she's an advocate of gene therapy. Yes, yes. Okay. So, yes. Now, biotechnology in medicine, there is one more thing. Early disease diagnosis. So we saw that how it was used. So one very important thing that was done was insulin production. Second thing that was done was gene therapy. And the third thing is that with the help of biotechnology, we can diagnose some diseases as well. Okay. For example, some methods that you already know is polymerase chain reaction. polymerase chain reaction, my dear children, it is responsible for basically finding out a trouble even if the level of pathogen is very less. So even the small amounts of pathogens, my dear children, can be detected using what? Using the polymerase chain reaction. So even the small amounts of pathogen, okay, suppose a small amount of pathogen DNA is there. If at all you use PCR, what is going to happen? You are going to amplify it. You are going to increase its number, right? Which is why you will be easily able to find even the small traces of pathogens. So there's a very famous question, what is the method that you use to find even small traces of pathogens? It is polymerase chain reaction, used for HIV detection, cancer gene mutation identification, and genetic disorder diagnosis. So HIV detection, we think it's always ELISA, but you need to understand that with the help of polymerase chain reaction, if at all we get to understand that there is a retroviral DNA out there, okay, and we are able to amplify it. So obviously you can find HIV also. So for HIV detection, for cancer gene mutation, for everything, polymerase chain reaction is the best. Okay, apart from that, what do you have? You have advanced technologies such as recombinant DNA technology. Okay, wait. Okay, then we have DNA, RNA probes and autoradiography. So radioactive probes hybridize with the complementary DNA. Mutated genes do not hybridize, allowing detection of, okay, just wait, listen to me very carefully, okay. So over here, suppose this is the DNA that is normal DNA and this one is a mutated DNA. That means it has some mutations. So generally, from the normal DNA, what did you do? You made a complementary strand. You made a complementary strand, right? And you tagged it with some radioactive thingy. Cool. So this particular strand is tagged with some radioactive thingy. Now what do? You make it and go and pair. So generally, the complementary pair should go and bind up there. And since it is radioactive thing, it will have that particular visibility over here. Okay, so if at all I'm putting it in an autoradiography machine and I'm viewing it, I'm able to see that the probe goes and attach over here. Why? Because it is just like a normal gene out there, it is complementary. It will not go and attach over here. Why? Because it does not have the complementarity. Does it have the complementarity, my dear children? No. Because it is mutated, which is why here this particular segment is not, the probe is not going to come and attach over here because it is not complementary to the mutated gene. That means that this gene is what? Mutated. So that is one method that we can use, and just give me a second. Keep. Yes. Okay. Okay. Got it? So radioactive probes hybridize with the complementary ones. Mutated genes do not hybridize, allowing detection using autoradiography. Got it? Yes, that's all. That's the second method. The third is enzyme-linked immunosorbent assay, which is ELISA. That is also a method used in biotechnology. Okay. And over here, what do you do? What is the basic funda of this? This is based on antigen-antibody interaction. Antigen-antibody interaction. Now, this has also been asked, what is the basic function or what is the basic principle of ELISA? It is basically antigen-antibody interaction. How does it work? Listen to me carefully. So over here, okay, suppose I have already got the antibodies from an HIV patient. That means I know that these antibodies are going to be produced if a person is infected with HIV. So these are ready-made antibodies that I have already. Now, there is a patient X, and patient X want to know that basically whether he or she has HIV. What do? We take the blood. In the blood, what do we have? We have antigens. If the person is affected, they will have the antigens. If the person is not affected, they will not have the antigens. Antigens basically means the pathogen. That means the HIV virus is some particle, either the DNA, RNA or the protein, whatever is there. Okay. So that is up there. Antigen is there, okay, of the pathogen in this particular organism. But how do we make sure that they have the antigens? So listen to me carefully. When the blood is taken and basically it has been treated, it is found that the antigens will come bind on the antibodies. Why will it bind? It will only bind if at all the antibodies are produced for these antigens only. That means they have to be HIV antigens. Because they are HIV antibodies, they will only bind with the HIV antigens. Now still you cannot clearly see it. So for viewing it, you have the next antibody that you put into the medium. This antibody will also go and attach, and linked to this antibody is an enzyme. This is an enzyme. What do? You add a chromogenic substance, a coloring agent out there. If at all of this is happening, the enzyme basically will be acting on the chromogenic substance and give a color change. When it gives a color change, we get an understanding that, okay, the antibodies were already there. This particular patient's blood had the antigens, which is why it went and bound there. And now when you put the secondary antibodies out there, they also went and bound there, which is why there's a color change out there. That means the person has HIV infection. So over here, antigen-antibody binding is the principle. You get it? And with the help of that only, we are able to find the disease. So based on antigen-antibody interaction, detects infections by identifying either antigens or antibodies. Similarly, my dear children, we can take the antigens already. We know the HIV antigens. Now we can take the patient. In the patient's body, obviously, the antibodies will be produced to protect the patients. Now, when you put that, the antibodies will come and bind, and the same thing is going to be repeated. So that can also be done. So either with the help of antigens, either with the help of antibodies, you can find whether the person has some trouble. Got it? So this is what I need you to remember. Very, very important thing. So polymerase chain reaction for finding even the small traces of pathogens. It can be used for HIV detection as well. Cancer gene mutations as well. Do not forget it. And DNA, RNA probes and autoradiography. This is best for finding the mutant genes. And enzyme-linked immunosorbent assay. Basically, the basic principle is what? Antigen-antibody interaction. Done? Done. And the last part, okay, there's a question. Which of the following diagnostic tools allows the detection of very low concentration of bacterium or virus by amplifying their nucleic acid? 2021 question. So by which of the method can you find very low concentration of bacterium or viruses by amplifying their nucleic acid? Is it ELISA or PCR, autoradiography, or DNA technology? Give me the apt answer. Think for it and give me the apt answer in the comment section. Okay, so please do that. Fine? Fine. Fine. Now the last part of this chapter, one more thing is there. After that it will end. So yeah, transgenic animals. I told you genetically modified animals are known as transgenic animals. Examples is transgenic mice, you have transgenic rabbits, you have transgenic sheep, cows and everything. But always remember, most of the times, the most famous transgenic organism that we use is a mice. So 95% of the case, it is going to be transgenic mice. Okay, we have so much things in common, we and the mice. Okay, now why do we use transgenic animals? What are the uses of transgenic animals? This is very important for your PU2. First one, to study of normal physiology as well as development. Suppose I know there is a gene, but I do not know what functions does it perform in a body. So what do? I take the gene and I put it inside the mice and I see what is going to happen in the mice. What is the effect on its growth and its development and the body function? You get it? So for understanding the normal physiology and development, or for understanding how does a gene work or what is the effect of a gene, you can use transgenic animals. It helps in understanding the gene regulation and effects on growth, development and body function. For example, insulin-like growth factor research. So insulin-like growth factor is a growth factor that is responsible for growth and development of a human body. You put it inside the mice and basically saw what are the effects that is happening in the case of a mouse. Okay, most of the times it is going to be having a similar effect in our body also. So insulin-like growth factors effect was being studied with the help of this, with the help of transgenic animals. That is the first thing. Second thing, for understanding the disease and for understanding the human model. So listen to me. We want to see that what exactly happens in cancer. Okay. What are the things that happens in cancer? So if at all we know that these are the things that happens in cancer, probably we can find medicines for treating those things as well, for increasing the longevity of an organism. So with the help of transgenic mice and other organisms, they created models, okay, and they studied diseases such as cancer, cystic fibrosis, arthritis and Alzheimer's. Ma'am, is the name of the diseases important? Yes. Cancer, cystic fibrosis, CC, cancer, cystic fibrosis. Apart from that, you have arthritis and Alzheimer's. So C square and A square, C square and A square. For studying these diseases and their effects, you had made models. Okay, now children who are watching this, obviously this is very grave to do to any other organism out there. Since they are not humans, we should not, it's not like we should not be considering them as organisms out there. But yes, this is how it worked. Nowadays, all of it is being still done, but the level of it has greatly reduced because there's a whole community to control all of this particular thing. Okay, we will be talking about that also. So second thing is to understand the disease study and the human models. Third thing, for production of biological products. For example, we saw that how insulin was being used. Okay, how insulin was being produced using E. coli. Similarly, my dear children, there is one protein, alpha-1 antitrypsin. Okay, so there is a gene in our body that produces alpha-1 antitrypsin. This particular gene or this particular protein is super important for treatment of a disease that is known as emphysema. It is a respiratory disorder. Okay. Now, if at all I want to produce this protein very much, obviously I cannot tell my body to produce it. What can I do is that take the gene and put it in some organism and let it produce, right? So similarly, that has been done. So used for human protein production, for example, alpha-1 antitrypsin for emphysema treatment has been produced using transgenic organisms. Okay. Second thing, Rosie, the transgenic cow. Why do you call a cow as transgenic cow? Again, it also has undergone some genetic modification. The name of this cow is Rosie. What was this particular cow producing? So this particular cow was producing milk, which was enriched with human protein. Human milk protein. So only in humans milk this particular protein was being seen, but now you took the gene, okay, which will be producing that particular protein, and you put it inside the cow, and the cow is now producing milk, having what? This particular protein out there, which you call as alpha-lactalbumin. So Rosie, the transgenic cow, in 1997 produced nutrient-rich human-like milk, which is having what? High amount of alpha-lactalbumin. So please remember, these two things are super important. Alpha-1 antitrypsin is a protein that is used to treat emphysema, a respiratory disorder. Okay, Rosie the cow, transgenic cow, in the year 1997 was made to produce what? Nutrient-rich milk, which was rich in human protein, that is alpha-lactalbumin. There is no 1 and 2 and things like that. Alpha-lactalbumin. Got it? This is very important. So for biological products also, you need transgenic animals. The fourth thing, for vaccine safety testing. So you know, on humans also vaccine safety testing was being done. But before that, my dear children, it's usually the animals on which we test the vaccines, and we see that how it works. So polio vaccine and everything was made to undergo the vaccine safety testing before human trials. Okay. Second, so the last thing that we have is chemical and toxicity testing. For example, an organism is made to be very sensitive. Okay, an organism is made to be very sensitive. Now in them we put the toxin substance, as in not toxin substance, basically a drug, suppose a drug, a medicine, and we are seeing that if an organism is this sensitive out there, and if we put this particular dosage of the medication, how is the organism going to react? If at all it is going to react badly, that means what? It's a problem. If at all it reacts normally, that means okay, this much dose could be fine. You get it? So for chemical and toxicity testing, genetically modified animals are used for quick and accurate testing of drugs as well as the other chemicals out there. So which is why we say that we should be having this drugs, this much mg of this particular medicine is okay. We cannot have too much amount, right? Why? Otherwise we will also die. So basically this has been tried, has been tested as well. Okay, so first is the uses of transgenic animals. The first is to understand the normal physiology as well as development using, example is IGF, that is insulin-like growth factor, that was being put inside the organism, and it was checked that how it regulates the gene and how it's responsible for growth and development. Apart from that, for disease study and human models, for example, for understanding cancer, cystic fibrosis, arthritis, Alzheimer's and all, you had some models being created. Okay, and now biological products, first is alpha-1 antitrypsin for treating emphysema. The second one is the human milk protein was found in the cow's milk, and that was the protein which was found was known as alpha-lactalbumin. It was produced using a transgenic cow, that is Rosie, in the year 1997. Yeah, then vaccine safety testing, and then chemical and toxicity testing. These are the uses of transgenic animals, and the most used animal is what? Mice. Always remember that. Now, ethical issues and biopiracy. Remember, whenever you do something like this, obviously there should be someone to control it. Otherwise, what is going to happen? We are going to kill all the organism out there. You know, world is not a good place to live, is what people say. Yes, so need for regulation. Unregulated genetic modification can have unpredictable effects on ecosystem. Sometimes some people can produce something that can be a bioweapon. We consider that coronavirus, which was being formed, also was a bioweapon, right? People probably could have made it as well. We never know about it, right? So the same thing is there. We need some regulation such that it does not have any unpredictable effects. And ethical concerns over the manipulation of living organism. You're dealing with a living organism. At the end, mice is also there. It's a chutku organism out there. It also has family, right? So we should be considering about the ethical concerns as well. That is why there was a need for regulation, which is exactly why there is a committee being formed, and that is known as GEAC. The full form of this is very important. It is known as Genetic Engineering Approval Committee. Genetic Engineering Approval Committee. So before doing anything, you have to get an approval from this particular committee. Before doing any genetical engineering, you have to take an approval from this particular committee. That's how you remember the name. Genetic Engineering Approval Committee, GEAC. Okay, approves GM research and ensures safety. Now, that is one part of the story. Now listen to the next part of the story. You know what happened? Basmati rice is found in India, right? But in America, in US actually, they patented basmati rice and said that no one can produce it. When you say that someone has got the patency, it means that no one else would be allowed to produce it. No one else would be allowed to sell it off. You get it? There are a lot of troubles when you get the patency of it. And don't you think that is a big problem? Yes. Why? Why is it a big problem? Because it is produced in India. It was something that is Indian. So obviously you need to take the permission from Indian farmers first. How can you put patency on something like that? Which is why some few things came up into the picture. So companies patent genetic materials and bioresources developed by indigenous people, as in the people of their actual origin. Okay. Example, in 1997, a US company patented basmati rice derived from Indian varieties. Okay, and this process is known as biopiracy. When someone does that, it is known as biopiracy. Unauthorized commercial use of bioresource and traditional knowledge without compensation. So whenever you're not compensating anything, whenever you're not getting monetary benefit or anything else, and you are saying that I am going to use all the bioresources up there and things like that, this is known as biopiracy. You have to talk to the indigenous people out there, the actual people who produce these products. Okay, yes. So developed countries, financially rich but poor. Okay, wait. So that is one very important thing. And as I said, this this process of when you say that you cannot, no one else can produce it or no one else can sell it, it is known as what? Patency. Okay, you call it patency. Now, against patency, you should understand that there is something that is known as biopiracy. Biopiracy is when some people, they commercially sell the bioresources and things like that without giving proper compensation. That is known as biopiracy. Okay, got it? So patency is different. Patency is like you're getting a right for something, and biopiracy is when you're doing it unethically. When you're doing it unethically, it is unauthorized. When it is unauthorized, you call this as biopiracy. Okay, yes. Now, preventing biopiracy. Some nations are creating laws to protect their bioresources. Indian Patent Bill Amendment includes provision for patents, emergency situations, and R&D support. There is research and development support. So what do we have against this? Indian Patent Bill Amendment was being passed that includes provisions for patents. Okay, that went ahead against all the organizations which were doing patency for no reasons and which were doing biopiracy for no reasons and things like that. Okay, so Indian Patent Bill was also being passed. Please remember this. I know this part is not very much clear, but just have the understanding. GEAC stands for Genetic Engineering Approval Committee. Without the approval of this, you cannot go ahead with genetic engineering process. Second thing that we need to have in our brain is what the term patency. Patency means you are getting the right to basically say that, okay, no one else can do any production or probably the selling point of things. No one else can be doing that. Now, if at all someone is doing it without in an unauthorized way and without giving proper compensation to the people from where you bought things from, basically this will be known as biopiracy. And to have all of this under control so that no one will do the patency by their own will, there is Indian patent bill amendment that was being passed, which controls everything out there. That's all. Okay, these are the few things that you need to keep in mind. So usually in your NCERT, all of it is like given in this manner. So I just made it in lines, that's all. So developed countries financially rich but poor in biodiversity. Developing countries, what is happening? They are biodiversity rich but vulnerable to exploitation. And India is a developing country. So it's our duty to safeguard our country. Yes, yes. That's all, my dear children. This chapter ends with this particular note. I hope you have understood what is the actual thing that you have to study in this chapter. Basically, everything is important. Every single bit of this particular chapter is important. This is not a very big chapter. It's a small chapter. You can just divide in your mind that biotechnology was used in plants, biotechnology was used in medicine, what are the things were there. Please have that in your head, solve questions. And as of now, Masterstroke live is definitely stopped for KCET questions. We'll be coming back after your examination. But for PO2, we have launched the Endgame series. Make sure you watch the videos. It is definitely going to help you out as well. Okay, so wishing you all the best. Please take care of yourself. Lots and lots of love. Bye-bye. If you have any doubts, put it down in the comment section. If you like the session, do let me know that as well. Bye-bye.